Good afternoon, ladies and gentlemen. I guess we can get started. I would like to start by acknowledging the traditional owners of the land in which this event takes place today, the Wurundjeri people of the Kulin Nation, and pay my respects to their elders, past and present. I'm Fernando Guimarães from the Walter Eliza Howe Institute. I also represent the Melbourne Immunotherapy Network, and I'm pleased to be your MC here today. I would like to just give a brief explanation about the VCCC. The Victoria Comprehensive Cancer Center, it's a powerful alliance that unites 10 different organizations in Victoria, which all aim to provide cancer control. And one of the systems that it's worked by the alliance is to stimulate collaboration between the different organizations and members and integrate research, clinical care, and teaching. And one of the contributions to the community is the provision of special lunch life series like that one, or trainings. On the Monday Lunch Life, we have like a couple of interesting talks from international and national speakers coming over to debrief about their experiences on their topics and talking about biomedical related hot questions at the moment, such as Professor Fran Boyle next week communicating the financial toxicity of cancer therapy, Caroline Johnson from Sydney talking about what's new in the law regulating health data, and Professor Nick Taylor on the last Monday of the month, which will be talking about a drama which is common in the life of any scientist, which is to publish or perish. But today, we have a special guest coming from the Monash Biomedical Discovery Institute, who is Dr. Julian Vivian. Julian Vivian has his laboratory focused on the structural biology understanding of the KIRs, the killer cell immunoglobulin-like receptors, which are critical factors to determine the immune response of the immune cells called natural killer cells to the target cells. It's what determines if they are going to kill or not. And they're particularly interesting not only in cancer immunotherapy, but also infection and transplantation. Some of the contributions of Julian, between many, they were accepted worldwide by recognition in top-tier journals such as Nature Structural Molecular Biology, JAXMed, JCI. Not only a few, but actually many Nature Immunology papers, which are really high-profile journals and very hard to publish in. I would like to thank you, Julian, to accept your invitation to come over, and we are all looking forward for your talk today. Please come along.
Well, thank you for a very gracious introduction, and thank you very much for the invitation to come down here and present my small corner of the world to this rather broad church of immunobiology. And welcome also to the online audience, which apparently is watching on the stream. I'm sure there's thousands of them out there. I hope you can all see me. Right, so today's talk will be focused on KIRs in cancer, which is kind of a rather broad title. More specifically, I'll be probably starting off on the sort of fundamentals of KIR, what they are and what they do, and then I'm going to sort of drill down into acute myeloid leukemia, so how KIRs can play a role there in hematopoietic stem cell transplantation settings. And as I mentioned in the introduction there, I'm a structural biologist. So I bring a rather different perspective perhaps to, let's say, a classically trained immunologist or someone who's coming from a medical background. So I'm not a clinician. So I perhaps have a different viewpoint. We have an hour here to luxuriate in KIRs. So if there's anything which you wish to add to the conversation, or if you want to interject with questions, please feel free. And here I've just put one of these word clouds there. They're quite a bit of fun, really. When you put in natural killer cells and KIRs, you get things like immunotherapy, tumor, cancer. What you don't get, but what should be there, is sort of Donald Rumsfeldian known and unknown unknowns, right? So some of these unknowns are simply because I perhaps don't know them myself, but there are certain aspects of NK biology which we talk about with a lot of confidence, but we don't really understand. So I'm going to mention things like education. So we can measure this phenomenon of natural killer cell education, but we don't really know the mechanisms behind it. And also jargon. So this is a jargon-heavy field, and I'll do my best to minimize that as much as possible. So this is a sort of natural killer cell and KIR slide 101. What are KIRs doing on natural killer cells? So here we have our KIR expressed on a natural killer cell, and the target cell expresses its ligand, which is a human leukocyte antigen, or HLA. And even in the presence of an activator signal, this interaction here is enough to inhibit the lysis of this target cell by this natural killer cell, the cytotoxic natural killer cell. But then in the absence of an HLA ligand, as occurs in tumors or in viruses, you get a loss of this inhibitory signal, you get disinhibition, and you get NK cell lysis. That's the fundamentals of missing self. If you miss the self ligand, you lose the inhibitory signal from the KIR. So what is this ligand? What is human leukocyte antigen? You can think of these as molecular tweezers that bind peptides. So these peptides are from the intracellular turnover of proteins, and they're presented on the surface by this molecule. In that way, it provides a snapshot of what's going on inside the cell. So something changes, say there's a viral infection, you're going to get viral peptides presented on the surface, and the immune system will see that as different. And in the case of KIR, if a virus tries to get rid of HLA, or a tumor tries to get rid of HLA to avoid that system, the NK cell will notice a lack of the HLA. So this molecule, the crystal structure here shows that it's a dimer. It's got this beta subunit here, beta 2M, which comes together with this green heavy chain subunit to form a cleft. Right? And in that cleft, we have the peptide in pink here. This is another view of the same molecule here, that the peptide which is presented to the immune system. So HLA are probably the most polymorphic molecule in the human genome, and these polymorphisms reside on the peptide binding cleft. So what this means is that across the population, people have different HLA, so they present different peptides, or they have different essential markers, and they have different outcomes in disease. So there's always a bit of an arms race between infections and the immune system, and the HLA is constantly mutating to keep up with this. So if you think about a classic missing self system, as I said, the peptides are presented for presentation to the immune system, and the classic receptor that recognizes this is a T cell receptor. So T cell receptor, is very peptide focused, and it triggers the cytotoxic T cell to kill any antigen that it hasn't seen before. And then down here, I've shown here in pink, are missing self style receptors. So Li49, so Li49 here, which is a murine equivalent of the human KIRs, binds under the cleft, as you would kind of expect a missing self receptor to do. So it's not really peptide focused. Same here, the LIRs. Again, they're just looking for presence, absence of HLA. They're binding pretty much in a monomorphic part of the HLA. They're avoiding all this polymorphic stuff here and the peptide. Again, looking for presence, absence of HLA. The one exception that sort of proves this rule is the CD94 NKG2A system, which is peptide focused. And it's peptide focused because it only looks for one peptide. It's looking for the peptide of the leader sequence of HLA class 1. So again, if a virus or a tumor tries to downregulate HLA class 1, we lose this one single peptide, the leader sequence to that. And again, our missing self will see that. So these receptors here are not particularly polymorphic. The T cell receptor, of course, undergoes massive recombination to generate many different versions of it to keep up with the peptide repertoire. So in contrast to these classic missing self receptors, we also have the KIR receptor here. This is KIR3DL1, comprises 3 domains, a D0, D1, and D2. There are also 2 domain KIRs. They have just a D1, D2, so they lose this extra binding enhancer, we call it the D0 domain. And the contrast here is, as a missing self receptor, it's actually binding over the peptide. All right, so this thing is not only binding peptide, but it's also binding this alpha-1 helix, which is a point of differentiation between different HLA classes. So it's actually looking for very specific HLA classes, and it's also dependent on peptides to a certain extent. And in keeping with that, actually I just showed you this other view here, it's very C-terminally focused. So it's only looking for P7, P8, and P9 positions of the peptide, but the peptide nonetheless. And in keeping with that, this receptor is highly polymorphic as well. So there's about 120 different versions of KIR3D1 currently annotated. Paul Norman, a geneticist, now at Denver, I believe, has estimated there's probably thousands of KIR3D1, almost as many KIR3D1 as there are HLA-B ligands for them. So there's about 4,000 different ligands of HLA-B, got probably a couple of thousand different KIRs potentially. This is probably the most polymorphic receptor-ligand interaction, excluding the T-cell, B-cell systems, which are, so it's the most polymorphic germline encoded system in the genome. Which raises an important question, which is why? Why would you want a missing cell system to be so dependent upon polymorphism? And I don't have a good answer to that question. Certainly there is a selective advantage in KIRs being tuned to their ligands. But whatever the true reason is, what it does mean is this polymorphism across the population results in different disease outcomes for different people. So this is really clear in diseases such as HIV. So HIV will downregulate HLA class 1. And depending if you have a very strong KIR receptor for that particular HLA in your makeup, you have a better chance of surviving HIV. And similarly, this sort of system can be exploited in HSCT or hematopoietic stem cell transplantation to improve patient outcomes, which I'll get to. So that was the 3D L1 receptor. But apart from being polymorphic, it's also a polygene family. So we can subdivide the KIRs into two different types. We've got our inhibitory KIRs here, shown in blue. And they include the 2DLs, 2DL1, 2, 3, and the 3DLs, 3DL1 and 3DL2. And as I said before, the only real difference structurally is that the 2Ds just have this D1, D2, whereas the 3Ds have this extra domain here. And as well as our inhibitory KIRs, which are our classic missing self receptor family, we have these activating KIRs. All right, so they don't give off inhibitory signals, they activate the NK cell. And exactly what they're doing is somewhat unclear at the moment. But again, they're made up of both 2D and 3D style receptors. So within the body, there's a huge diversity of natural killer cells. So this paper here, looking at NK diversity using CyTOF, looks, I think, at 28 markers, including the KIRs. And per individual, they're finding about 30,000 different NK cell types, of which they estimate, again, there's probably 100,000 different NKs across the population. And of these, maybe 50% express KIR. And again, the KIR are clonally expressed, so any one particular NK clone might only express one KIR or two KIRs. So we had this huge diversity in the NK repertoire. And what this also sets up is that there is a pool of NK cells that are either educated or uneducated based on their KIR profile, which is important for transplantation settings. So this is a simplified view of what education is. Here we have our autologous cell and we have our natural killer cell here. If you express your cognate KIR HLA pairing, during the NK cell will be educated. It will know what self is, and it's then primed to activate. So if any sort of loss in HLA expression here, the NK cell will trigger. The alternative is the other subset of NK cells, which are uneducated, right? They don't express any KIR. So even in the absence of HLA, they're not going to react, right? So their threshold for activation is much higher. And this gives us targets for therapeutic intervention. So the first one, of course, when we have educated NK cells and they're in the presence of their ligand, we have essentially a lack of missing self, which provides us with an opportunity to perhaps interfere with this system to trigger lysis. Similarly, with our uneducated cells, so they may not be sensitive to HLA response or expression. They are very sensitive to CD16 antibody binding. So uneducated cells and educated both express a CD16 low affinity Fc receptor. But in terms of responsiveness, it's actually the uneducated cells that are more responsive to CD16 ligation. And again, this is another point where therapeutic intervention can take place. So you can try to dampen this KIR HLA response to make it more likely to trigger a ADCC style response. So a takeaway summary so far from the background: the KIRs are a multi-gene and highly polymorphic family of receptors, and the inhibitory KIR educate the NK via their interaction with HLA, and that determines the responsiveness of the HLA to its environment and its self-tolerance. Activating KIR are also implicated in education. Generally, they educate in the opposite way to inhibitory KIRs, but this is less known. And this education process can be targeted for therapeutic intervention. So what do we want to know, right? So how do we go about trying to target this system? Essentially, what we want to know is what makes a good KIR HLA pairing, right? So we're starting pretty much at square one. And our method of choice for working this out is we use a Luminex-based assay. So we use commercially sourced beads, which have 100 different HLAs on them. So we're sampling across the thousands of HLA that there are, looking for the most prevalent ones in the population. And these are fluorescently tagged. And then we have our fluorescently tagged KIR. And essentially, we're just looking for a co-signal of KIR and HLA in our Luminex screen. And the benefit of using this style of approach is the HLA in these beads is taken from cells. So it's got a broad repertoire of peptides, so our results should be peptide independent. And the basic workflow is, we want to identify ligands. I want to put them in a structural context because I'm a structural biologist. The clinicians and immunologists mostly want to put these into disease association studies, right? So we're trying to clinically, or put these into groupings that are clinically relevant. So as I said, the KIR are highly polymorphic, but you can kind of divide these into broad lineages. As I said, there are 100 currently annotated. There's likely to be more than 1,000. So how these lineages hold up over scrutiny of the broader range remains to be seen. We're excluding the activating KIR through S1 for the moment, but we have sampled across this 002 style group, 001, and 005. And we put these through our Luminex screen, and we're looking for reactivity to HLA. So of the 3 allotypes we tested, what we can see is actually there's differences in binding to HLA. So down on this side, we've got all the HLA B, W ligands, right? Segregated by either isoleucine, adposition 80, or threonine adposition 80, which I'll get to in a second. And this is a rank score of binding. So this is not an MFI, but it's a rank score. And 2 things that stand out straight away is that they're not the same, right? So this one here, KIR 005, can recognize, look at it on this one, sorry, it's too close to me. B4403 rather well, and A2402, whereas these are not well recognized by the 001 and 015 lineages. So we look across the HLA-B landscape. What we're saying is that KIR are binding HLA-BW4, which is well established, and which is found mainly on HLA-B, but also on HLA-A. And the ones that's not binding are BW6. This is a nice system because essentially it's either binding or it's not. There's no cross-reactivity between BW6 and BW4 for the KIR 3DL1s. So what is this BW4 motif and why is it important? So the BW4 motif is found here on the alpha-1 helix right there underneath this D1 domain of the KIR. It's a short stretch from position 77 to 83, and that defines the lineage of HLA-B. The BW4 motif itself is somewhat polymorphic, just shown here different BW4 motifs. And the one position that seems to be important is this position here, position 80. So this is kind of an historical dimorphism that's important in HIV. So the isoleucines at position 80 are considered the strong binders, whereas the threonines at position 80 are considered the weak binders. So the isoleucine 80s are associated with non-progression of HIV infection to AIDS, and the threonines and BW6s to rapid progression to AIDS. But when we look at our data, that simple dichotomy doesn't necessarily hold. As we can see here, there are some threonine at position 80s that do bind better than some isoleucines at position 80. But generally speaking, the isoleucine 80s are the better binders. So as a quick categorization, it actually works fairly well. So how can we exploit this to improve HSCT, hematopoietic stem cell transplantation, in the setting of acute myeloid leukemia? So a long time ago now, 16 years ago, Rubieri and others showed that if you mismatch the donor NK cell with the recipient HLA, you get improved outcomes in AML. So essentially, if you take an HLA with a BW4 isoleucine 80, these are the strong inhibitors, and you put that into a recipient that has essentially a weak ligand for KIR, you're going to essentially trigger missing self. And the way that this work is classically categorized is a weak ligand is HLA BW4 threonine 80 or BW6, like here. And a weak KIR is a KIR 3DL1 that's low surface expression, whereas a tight one is a high surface expressed KIR 3DL1. And so Boudreau and Su have gone and quite elegantly shown that if you do similar, this sort of donor-recipient mismatching, you can get improved outcomes in AML. So there's been a long story between Rubieri's initial studies and this study published this last year. And most of that revolves around how you condition the NK cells and other treatment regimes, but the most important factor seems to be T-cell depletion. So you need to T-cell deplete the graft before transplantation, and this results in the better outcomes. But all things considered equal, KIR allotype and HLA allotype makes a difference. So how they stratified their study, they took strong KIR 3DL1 receptors as being high surface expressed and weak as being low. Poor ligands as BW6, of course, and isoleucine 80, and threonine 80, and the strong as isoleucine 80, as we discussed before. And they made these categories here. So non-interacting pairs, weakly interacting pairs, and strongly interacting pairs, with the strength of this graft versus leukemia effect going in this direction. So what you're looking for here is the donor NK cells, the graft, triggering an anti-leukemia effect on the recipient. And so this is what their data looks like in a nutshell. In the yellow line and grey lines here, we have non-inhibiting and weakly inhibiting pairs. The probability of relapse for their patients over time in years. And as predicted, it's these weak and non-interacting pairs that have a lower incidence of relapse, and it's these strong KIR HLA pairings that have the greater incidence of relapse. Similarly, when we look at survival versus time, again, in green, in yellow and grey, we have these weakly interacting pairs having a higher probability of survival than the strongly interacting pairs. And then when you subdivide that data into sort of relative hazard for relapse by actual type, so KIR321 low, IADT, IADI, sorry, isoleucine 80. What you can see is that, okay, the non-interacting pairs of BW6 have a lower relative hazard for relapse and overall more mortality. But then things get a little bit different with this sort of weakly interacting 80 threonine categorization. So the low here with ADT should be a relatively weak interaction, but actually you have quite a high hazard for relapse. And as expected, the isoleucine 80s are really strong guys. Strongly interacting pairs have a high incidence of relapse and mortality. So this data point here is quite a curious one, and does suggest that although this system does work and categorizing the HLA and KIR pairings in this way is going to work, there's gremlins in the data as possibilities to improve this system. So to take away a central message from this rather nice study is that KIR321 HLA donor recipient mismatching is therapeutically benefit in HSC treatment for AML, right? But the current clinical trials, actually I should point that out. So based on the strength of this data, this sort of classification is now being used in a clinical trial. As I understand, it's an observational trial, and when all other categories are met, the treating physician can choose whether to add this as an extra factor for their patient. So the current clinical trial is predicated on this KIR high-low BW4 isoleucine 80 stratification. The question is, can this stratification be improved upon? And if you remember our initial data with the binding data, and that would suggest that yes, we can actually improve upon this. These classifications, so they are, and I should say this, I don't want to take anything away from this study. These classifications are mostly historic, and although they will work across sort of broad groupings of patients, on an individual basis, they can probably be improved. So I could probably spend an hour going through just this table alone, but I just want to point out a couple of points. So talking about high-low, 001 and 015 are the classic high expressors. And even in this system, we can see that some isoleucine 80 BW4s don't bind very well. And some threonines are much better than the isoleucines. Similarly, this classic low expressor 005 actually binds isoleucine and threonine alleles rather well. All right, so this simple dichotomy doesn't actually hold true, at least for these three that we've tested, and we assume this is going to be broadly applicable across all the KIR321s when we start looking. And functionally, this is also true. So when we look at different lineages of primary NK cells and how they react to HLA expressed on 221 cells. So 221 cells don't express native HLA, but we can transfect HLA of interest. What we see again is that against normal 221, we have high CD107 expression, which is a marker of activation. Similarly for A24 and our BW6 here. This holds true for other alleles, and then we look at our classic low. The lower here actually binds all of them very, very, very well, and will functionally is reactive to all of them. So the obvious thing is put your money where your mouth is. If you're saying that you can do a better stratification of the database simply on binding strength, why don't you do it? So this is something that we've been trying to scrape together the pennies to do the high resolution typing for, and so far we haven't managed to get the funding, but hopefully soon. But in the meantime, we've done an analogous study in HIV, where our collaborators have access to large cohorts. So just to summarize a lot of data here, what we're doing here in HIV is you want a strong KIR HLA pairing in order to maximize chances of survival. So these plots here are looking at viral load versus days of HIV, and similarly T cell count, and it's stratified here based on our binding data, solely based on our binding data. So blue is the 50th to 100th percentile, and red the 0th to 50th percentile. And as you can see, for the tight binders in blue, we have a really low, also we have a lower HIV count than with the low binders. And similarly, our CD4 counts in this HIV model are higher for the higher binders and much lower for the lower binders. And we stratified that by actual individual KIR3DL allotypes, so 001, 2, 4, 5, 15. Again, it's the same pattern for viral load and for CD4 T cell count. And in terms of a prognosticator for how well you're going to do in terms of CD4 T cell and HIV load, this is so far the best predictor that we have on a genetic sense. So the HIV people are very excited by using this binding data as a predictor of outcome. So how we would improve this mismatching 3D1 strategy in AML is doing the same thing. So we would get rid of this isoleucine threonine dichotomy, we'll get rid of this high-low KIR3D1 expression pattern and focus solely on binding strength. So we'll take 3DL1 HLA high binders and move them into patients which have lower binding strength. So it's a pretty simple model. And what some of you may have noticed also in the Sue Boudreau study is they're only really looking at KIR3D1 type from the donor and HLA type from the recipient, and somewhat ignoring the HLA type from the donor. So what we actually want is highly educated donor cells. So we want to look for the most highly educated donor cells and then putting those into our recipients. This is also probably important for adoptive transfer as well. So if you're going to expand NK cells ex vivo to put into patients, again, you want the most highly educated NK cells to mediate this graft versus leukemia effect. And we would advocate that this should be done on the binding strength rather than more historic models. So the takeaway message here is that the BW4 containing HLA differ in their ability to inhibit target cell lysis by 3DL1. These differences are not entirely explained by the currently understood isoleucine 83-amine dimorphism. 3D1 allotypes also differ in their ligand recognition preferences. And again, the high versus low model doesn't necessarily correlate with inhibitory potential, right? So our HIV study suggests that mismatching as per the Boudreau-Sue model, although this will work, will be improved if we use binding strength as a stratification criterion. Running for time. Right, so I just want to spend the last few minutes talking about leronlimab. All right, so leronlimab was founded by this company, Innate Pharma, and their goal was to mimic this graft versus leukemia effect with a therapeutic antibody. So what they want to do, or what they have done, is made this anti-KIR, which is targeting the 2DL inhibitory KIRs, and the idea is to block this interaction and improve leukemic clearance. Right, so this is a history and importance of leronlimab to the field. So leronlimab was founded by people including Eric Vivier, who founded this company, Innate Pharma. Here he is here, and I've taken this from a talk he's got posted on YouTube. The talk is all in French, but the slides are in English, so you can get something out of it. Eric Vivier has strong connections to people in Melbourne, especially at the WeHi, but I don't have any connections here or a competing product. So I'm very much an outsider looking in on the leronlimab story, and I'll give you some perspectives there. So this is just a timeline of leronlimab and KIR. So from the initial discovery of KIRs in 1993, in 2005 they have a drug candidate. So it's pretty rapid progression. They validated it in 2005 in transgenic mouse models. It should be noted that mice don't have KIR. So KIR is unique to the primates. So what they did is they transduced or made a transgenic mouse with a single human HLA and a single KIR, and that looked efficacious, but of course it ignores some of the complexity of having 15 KIRs. And then in sort of 2006, they signed an almost $500 million deal with Bristol Myers Squibb. So for the small niche KIR field, this was a huge splash, and it's hard to sort of overestimate the importance this was to the field. It really put KIRs on the map, and from there they moved into phase 1 or phase 2 clinical trials. So what happened? Of the 9 trials that they've started, 7 have already been abandoned as they've failed. So far it's failed every phase 2 clinical trial. It's just not efficacious. This includes with PD-1 antibodies and other combination therapies. So the question is, what does this mean for anti-KIR mAb development, and what went wrong? So as big a splash as it was to get lorilimab up, its failure has also kind of sent ripples through the field. And you will hear people say that KIRs just aren't that important in cancer and immunotherapy. Perhaps that opinion's a little bit premature. So what you may have noticed from their rather simplistic model of how they think lorilimab is working is that it's targeting the inhibitory KIR2Ds, which are 2DL1, 2DL2, and 2DL3. And they bind HLA-C. So unlike the KIR 3DL1 system, where you just have a single KIR binding BW4, you've got 3 KIRs here, and they bind all the HLA-Cs together. So there's a lot of crosstalk and cross-reaction between them. And perhaps what's not entirely appreciated, because it hasn't been published, is we've done our same binding study, Luminex-based binding profile study for the activating KIR. And they also all bind HLA-C. And lorilimab binds at least 2DS1, 2DS2, perhaps 2DS3, and maybe 2DS5. All right, so you have a monoclonal antibody that's targeting a highly complex system of 8 separate receptors with overlapping affinities for a single ligand family. I'll take a step back here and I'll talk about what an activating KIR looks like relative to inhibitory KIR. So we saw the structure of 2DS2 in complex with HLA-C7. So the activating KIR is shown here in this sort of salmony color. Here's the HLA and the peptide, and its inhibitory counterpart is this green line here. And all you really have is a slight dislocation of this D1 domain. And the only strong polymorphism between the two, between activating and inhibitory, is this Phe to tyrosine substitution in position 45. That's a single hydroxyl substitution. Right, so you can switch the affinity or the binding strength of these receptors by switching this one hydroxyl group. And this distance here, 1.5 angstrom, is exactly the dislocation of this D1 domain. And all this does is it makes it a weaker binder. Right, so the activating KIR bind the same ligands, but they bind them slightly weaker. So what does this mean in terms of lorilimab and education outcomes? So this is what they wanted to do, right? They wanted to target these educated cells with a monoclonal antibody and get this missing self response. Hasn't worked. One thing to note, of course, is that in our AML studies that people have done, looking at donor-recipient mismatching, is it only really works if you deplete the T cells. So this is putting a monoclonal antibody into T-cell competent patients. So looking at some sort of CD4 depletion might be interesting here as well as a way to rescue the drug. And a similar way to get around Tregs may be to infuse with IL-15. But I don't think that's been approved for studies or for patient input yet. So this is what they've desired to do. But what have they actually done? That's a big question, right? So you're targeting inhibitory and activating receptors. What have you done to the education of these cells? Have you actually essentially made this a hyper-educated but satisfied by antibody cell that's not going to react? Or by targeting, so it's hyper-responsive? Or have you made this guy here? Have you essentially messed around with the education system and now it's not educated on HLA at all? My guess would be that's what lorilimab's doing. Right? I haven't got the data to back it up, but as a hunch, that would be interesting to look at. And if so, there may be scope to look at lirilumab in the context of ADCC and rituximab. So of the 9 clinical trials, 7 have been abandoned. There's still 1 ongoing with rituximab. So there is potential that essentially they've created an uneducated NK cell pool, and there may be ADCC responses. So the takeaway message, I think, from lirilumab is that we have a hugely complicated system, and perhaps the instrument that they're using to target this is just too blunt. So a more targeted, specific anti-KIR may be more beneficial. And then looking at particular patients, perhaps it's good to type and look for a drug that's specific for particular HLA-KIR interactions in the future. And it's too soon to write off the potential of anti-KIR, I think, on the failure of a first-in-class trial. Right? This is the first anti-KIR available. And I would suggest that lirilumab's clinical failures are more a sign of a lack of understanding of NK biology than a sign of the lack of importance of KIRs. And I put that in there because I have heard this message from several researchers. So I think it's a watch this space message. Which brings me to the acknowledgement. So as I said, I'm a structural biologist based at Monash, so we're under the broader Jamie Rochon umbrella. A lot of the work was binding assays was done by Philip Pym. We have collaborators at the University of Melbourne, especially the Andrew Brooks lab, with able help from Philippa Saunders and Joey O'Connor, who's now moved on. The HIV work is a collaboration with the NIH, so Mary Carrington's group and Vivek. And I have to thank the Red Cross Transfusion Service for providing those 1 lambda assays that we use for our binding studies. And thank you very much for listening.
Thank you so much for this amazing talk, Julian. We have like a little present for you from the VCCC.
That's very kind. Thank you.
Hope you like it.
That's very, very kind.
I believe we have like a few minutes of questions. If somebody has anything they would like to ask.
I enjoyed a very interesting talk. So my question was, is it possible to identify a spectrum of education using quantitative? Is it possible to say, well, this cells this much educated, this cells even more educated?
Yeah, so our collaborators, Andrew Brooks, this is exactly what they have set up. They have licensing assays. I haven't got any data to show on that. But certainly there is this idea of tuning in NK cell responses, that the level of education is dependent on the level of HLA expression, and that this will actually change.
On the development of the educated NK cells, is it known what factor makes that start expressing here along the development?
During development? Oh dear. So I don't know what the triggers are. So certainly you can induce a memory-like KIR population with cytokines, but I don't know the actual order of events or exact developmental trigger, but it can be induced ex vivo.
Another question? Sandra?
Right, so just to pick up on your last point there, it sounded like one of the issues with the antibody therapies were KIR. So is it possible to get specific antibodies to subject to KIR so that we could use to match your antibody therapy?
So certainly people have made KIR specific antibodies. None of them are in clinical trials that I'm aware of. I don't think anyone's even perhaps thinking about that now, given lirilumab's disappointment. It's a lot of money to spend, and it's kind of a what's the word I'm looking for? Essentially, you lose the opportunity. It's an opportunity cost. That's what I'm trying to say. It's an opportunity cost to go after a potentially dud KIR antibody if you've got another checkpoint inhibitor that's potentially more profitable. But yes, you can make mAbs to specific KIRs, and I'd be interested to see how they work clinically.
And something that is quite hot in the field nowadays is the understanding of the possibility of shedding of CD16 out of the surface of the empty cells.
Yeah.
You showed a slide showing like a little similarity between SC receptors and some of the KIRs, I believe, in the beginning.
Yeah, so they come from the same lineage, right? They're Ig-based receptors, 2 and 3 domain. So there are structural similarities between the FC receptors and the KIRs. Yeah, the shedding phenomenon is a really interesting one. So I think there's a nice study looking at NKG2D ligand, the McCain-McB shedding, and how that blocking that with antibodies has proven very beneficial.
Can the shedding happen on a KIR receptor?
I don't know, actually. Certainly HLA can be shed, but I don't know if anyone's looked at KIR shedding. No, that's an interesting point.
Another question? Any students experience about something? All right, so